Welcome to the Diabetes Bio Podcast, a podcast for the Diabetes Journal, the flagship journal of the American Diabetes Association. Each month, we will interview one or more of the authors of the feature articles, the papers of the month, or other articles published in that month's issue of the Diabetes Journal. We also have a Sweet Talk segment of the podcast, which is more of an open format discussion of additional articles, conferences, or major events in the field that we'd like to highlight. My name is Darlene Sandoval, and I'm joined by my podcast co-hosts, Dr. Kirk Habiger and Dr. Kevin Williams. Hey guys, how are you doing today?
Good. Happy Tuesday again.
Happy Tuesday.
Hello.
Can you believe it's Labor Day coming up? That means the end of the summer.
Oh yeah, summer's gone. It's done. It's still hot, but
Yeah.
Yeah, I'm ready for that heat to go away.
So do you guys have a favorite summer moment?
The American Diabetes Association meeting was pretty phenomenal.
Nice.
I'm gonna go non-science. I had a bear encounter this summer.
Oh wow.
Where?
Yeah. It got in our car and tore it up pretty good. So that's a fun story that I'll have for a long time.
Wow. Where were you at?
We were in the Gatlinburg area, so up in the Smokies.
Wow.
Were you in the car?
No, we're luckily in the house.
All right. Well, Kirk, why don't you tell us what we have in stock for our interviews today?
Sure. So today we're joined by corresponding author, Dr. Teresa Ku, and lead author, Dr. Jose Ortiz, talking about their paper, Trefoil Factor 2, expressed by murine pancreatic acinar cells, is required for the development of islets and for beta cell functioning during aging. This work was done while Dr. Ortiz was in Dr. Ku's laboratory at the City of Hope's Arthur Riggs Diabetes and Metabolism Research Institute, where Dr. Ku is an associate professor. Then we had the opportunity to chat with Dr. Adrian Bella, professor of medicine at Mayo Clinic in Rochester, about his new paper, The Longitudinal Effect of Diabetes-Associated Variation in TCF7L2 on Islet Function in Humans.
All right. And Kevin, what's happening on our Sweet Talk segment?
Today we're highlighting the work of Dr. Matthew Weber from the University of Notre Dame. Dr. Weber received the Pathway to Stop Diabetes Accelerator Award in 2019 on his work on materials that can sense and respond to changing glucose levels. This research holds a lot of potential to help control blood glucose levels more effectively, which is important for managing hyperglycemia and hypoglycemia in diabetes. And while this research is still in the preclinical stages, I'm really looking forward to seeing how this research evolves and the impact it will have. So stay tuned.
Oh, that sounds cool. I also want to remind everybody that last week we dropped the release of our live podcast that we recorded during the ADA scientific sessions this June. This was a unique opportunity where we had all of ADA's podcast co-hosts together doing an episode. So in addition to the three of us, we had hosts from the Diabetes Core Update, the Diabetes Care On Air podcast as well. And it was recorded live in front of an audience even, or at least a few people were there. And it gave us a chance to discuss the highlights from this year's meeting. So it was pretty fun. It was fun to
Woohoo.
do. Did you guys enjoy that live podcast?
Yeah, it was really fun.
It was good. A little bit nervous in the beginning, right? But we got over it. I think we got rolling, and it was a good conversation.
Yeah, it was fun. I listened to it last week and was really surprised how well it came together.
Credit to the production staff.
That's right. Credit to our amazing production staff. All right, well, let's dive into the science.
Next, we're going to discuss a feature article from the September 2024 issue of Diabetes, which is titled Trefoil Factor 2 expressed by the murine pancreatic acinar cells is required for the development of islets and for beta cell functioning during aging. Today, we are joined by corresponding author, Dr. Teresa Ku, and lead author, Dr. Jose Ortiz. Dr. Ku is an associate professor at City of Hope's Arthur Riggs Diabetes and Metabolism Research Institute. Dr. Ortiz is a former student of Dr. Ku's and is now a postdoctoral fellow at the Koch Institute for Integrative Cancer Research at MIT. Welcome, Jose and Teresa. Congratulations on your paper, and thanks for being a part of this month's podcast.
Thank you for having us.
Yes, thank you very much.
So we like to get things going with a little icebreaker question. So the question I have for you guys today is to give us your favorite fun fact about science. So Teresa, we'll start with you.
That's a very difficult question.
There's too many to name.
I really cannot think of the on the spot. Maybe Jose can go first.
I think for me, a fun fact about me as a scientist is that I'm actually originally a painter, and that's how I got into science, like through art. Because being an immigrant from Mexico, coming to the US, I didn't know English, so I didn't understand text, but pictures and visuals were easy for me to pick up. So from the beginning, as a child in the US, I just gravitated towards numbers and pictures, and biology was very visual, so that's how I got into science. But I'm originally like art, so drawing, and I try to incorporate all that stuff in my work because I think it's fun.
Well, that's great.
That's really cool. Did you come up with something, Teresa?
Well, I can think of clearly happened during my childhood. I was a little girl and I was always asking my mother about something about nature. But my mother wasn't a scientist, so she just bought me a series of books to just shut me up so I can just read it and understand on my own. Yeah. So that's something that I was quite early on that I knew I want to become a scientist.
Awesome.
All right. So, Teresa, we like to give our authors the opportunity to highlight any other contributing authors or collaborators before we get started. Take your time here. And is there anybody who has contributed to this work?
Yeah, definitely. The work is a collaboration between my lab as well as the labs of Debbie Thurman and Patrick Fugger. So Debbie has, her lab has contributed to many of the phenotypic characterization of the islets that Jose was studying. And Patrick himself is an expert in the triple factor family of proteins. He himself has studied triple factor 3 earlier. So it was a natural combination of the three labs to explore the role of triple factor 2 in the pancreas.
Great. So now moving on, can you give us an overview of your research program and then maybe how that led to this specific project?
My lab has been interested in studying whether or not there are adult pancreatic progenitor cells. And that led us to study the ductal cell compartment because we found that the ductal cells within that population, there is a subpopulation that behave like progenitor cells. So we will focus on that for a very long time. And of course, we are also interested how different environmental factors that may affect these ductal progenitor cells. So that led us to looking at different secretory factors, growth factors, or ECM proteins. And triple factor 2 was part of that journey while trying to figure out what's going on in the pancreas. And quite to our surprise, originally we hypothesized that triple factor 2 would be an endocrine factor because the earlier study that publishing showing that it's endocrine pancreas that express triple factor 2. But we later found out that, with Jose's elegant work, that the astronaut cells are the cell type that express this particular protein. And I guess the rest is the history because now Jose has this beautiful work.
Definitely. So, Jose, why don't you tell us about so maybe do you have a little bit more information on TRFO factor 2? And it sounds like there's a bit of developmental expression that changes from, I guess, from development through adulthood. Maybe comment about that.
Yeah, so triple factors are very interesting proteins. There's only 3 of them, thank God. Um, and I was able to look at their expression pattern in the developing pancreas at critical time points during development as well as post-birth and adulthood. But the one that stood out was TFF2 out of the all 3, 1 to 3. TFF2 was highly expressed at the gene level, and then later at the protein level was detectable and expressed in the astronaut cells later when I did co-immunofluorescence staining with other markers. But TFF1 at the gene level is not really expressed in the pancreas at any time stage. TFF3 is expressed in the pancreas during development and in some of the endocrine cells early on. And then later they become specified to the islets, but the islets appear to not express any TFF2. So these are the expression patterns that I understood. And actually, TFF2 has been well studied, as well as the other TRFL factors in the gastrointestinal tissues. So I didn't have any other reference besides the intestines where the TRFL factors have been well studied and understood more in the context of injury and regeneration. And so I was really inspired by the intestinal field as my initial learning tool to then see how those findings in the intestine could apply to the pancreas. But it looks like in the pancreas, TFFs are playing a different role. So that's really interesting. But it was really important to figure out the expression pattern because two groups in the past have done the expression pattern analysis, and one group claims it's in the islets, and the other group claims it's in the exocrine tissue. So we were kind of both confused. And considering a lot of the folks didn't use the same antibodies, we wanted to use one commercially available that could be tested by various groups. And we later confirmed it was indeed coming from the exocrine cells, and in particular, the acinar cells are preferentially expressing this protein.
Nice.
As early as embryonic day 15.5 to 16, which is very specific.
So that's great. I actually want to dig in a little bit deeper there. Dr. Koo, I think a strength of this article was your use of multiple confirmatory studies to really interrogate the TFF2 system. Can you give us a little insight into that experimental strategy?
Well, as Jose just mentioned that initially we want to clarify the expression pattern in different compartments of the pancreas. So that would be critical. And Jose has been really great at testing multiple different antibodies, including those antibodies that has been utilized by previous publications. Another strategy that we really want to explore would be using genetic deletion strategy, because that would be something that will allow us to really interrogate what is the role of TFF2 in a physiological context. So my lab generated, we commissioned an Australian company to generate the TFF2 flux mice in the B6 background. And then we subsequently study that and then cross that with the PDX1 Cree mice so that we can have a pancreas and duodenum specific deletion of this particular protein and then characterize subsequently. So most of the work is published, presented in this diabetes paper.
Great. And you mentioned that you used the PDX Cree and you had duodenal and pancreatic deletion, right? Any caveats there that you want to discuss and maybe any plans to knock out TFF2 in the acinar specifically?
Yeah, Jose, you want to comment?
Yeah, so early on, I used online databases to look at the RNA expression pattern of TFF2 in the pancreas during development using the LINS database online, as well as Tablia murris for adult pancreas cells. And we found that there are some other pancreas compartment cell types like the endocrine progenitor cells, other endocrine cells that could express low levels of TFF2 mRNA, as well as the adult ductal cells that could express low levels of TFF2 RNA compared to the acinar, which express much higher. So we were concerned about potential compensation effects from the other compartments if we only use an acinar like mouse model to knock out TFF2 in the acinar cells. So because we wanted to avoid those potential confounding effects that could come from compensating cells, we just wanted to knock it out of the whole pancreas just to begin with. Using PDX1 Cree is a great mouse model that's well established. We use the one from Pedro Herrera's lab, which is great. Has really good recombination, Cree recombination. So yes, but that's the goal. Now that we know it is affecting the pancreas, now we can dissect specific lineages more carefully, but keep in mind that compensation effect that needs to be carefully looked at to and consider.
All right, we'll go back to Teresa and talk a little bit more about these findings. So using the RNA-seq, you found, and you guys have already touched on this a little bit, but you found that this protein that you think is expressed in acinar cells is affecting the beta cell pathway specifically. And so can you talk a little bit more about those findings and what your next steps were after that particular finding?
Originally, when we saw the RNA-seq data, we were not trying to expect anything, but to look at the data. We followed the data, and the highest downregulated pathway was pancreatic beta cells. So that got us quite excited. That may suggest this exocrine or the acinar cell secreted factor may affect the beta cell development. So Jose subsequently follow up with that and look at not just beta cell, but also other endocrine cells. And he found that the whole endocrine cells were affected at 16.5 pancreas, but not affecting the other compartment, meaning the exocrine compartment was intact. So that was interesting. And we also expect that because of decrease of beta cell numbers in the embryo, we also expect to see diabetes phenotype soon after birth, but we did not see that. So it was quite puzzling. So that was the beginning of the, I guess, investigation, and quite an interesting phenotype.
And then I guess as a follow-up question, if I can, how are these mice compensating for the lack of TFF2 and not becoming overtly diabetic?
The initial thought was that there could be some compensation coming from the other families of the triple factors. So we naturally look at TFF1 and TFF3 in the pancreas. And we found that TFF3, but not TFF1, was upregulated as a result of the TFF2 knockout. So we follow on that phenotype, and then Jose also painstakingly looking at different stages during the mouse development, newborn, and also soon after newborn, and also in the adulthood. And we found that TFF3 is not compensated until day 7 after birth. And that's another puzzling fact to us. Yeah, so why so late? And then he worked backwards and look at P0, the newborn, and didn't really find compensation. So in terms of TFF3, but by the time TFF at the day 0, the endocrine cell has already rebounced back to the normal level. So that was something that suggests to us that TFF3 compensation is not the primary driver, rather is some kind of a secondary effect from the early knockout of TFF2 in the pancreas. So that's another interesting fact.
Jose, now I have a question for you. You carried these studies well into adulthood, right? Assessing mice as late as 60 weeks of age. How did you land on that time point? And then tell us about the role of TFF2 in the aged mouse.
Yeah, so that was an interesting phenotype because when we allow the animals to reach birth, we would expect them to be overly diabetic because they show the developmental problems with the islets, but we didn't. And we later let them reach 6 weeks of age and they're okay. I did the glucose tolerance test, no problems, body weight measurements, no problems. I even dissected out the pancreas and looked at it. I didn't see any significant problems just by eye. But then we thought about what if we challenge these mice to see if there's an issue with the beta cells. So there were a lot of options, high-fat diet exposure, like STC-induced injury. But the other one was just aging. And aging to me sounded the most easy one to do because you just let the mice age. And age is a form of stress that could give you a phenotype. So we decided to go with aging because it's the one that would introduce less variables. And so, as I let the animals age, I painstakingly just like literally monitor their lives, and I would do the glucose tolerance test every time they reach like 20 weeks to 30 weeks, 40 weeks. And at 40 weeks, I started to see some differences. And then at 50 weeks, I started to see that difference still more. And by 60 weeks, I started to see a very clear difference between the 3 groups, the control, the heterozygous, and the homozygous pancreas knockouts. And that's where led me to see this difference. Yeah, there's a difference in the glucose response, so maybe we should go more in depth. And then that was my next goal, to figure out, okay, if there's issues with the beta cells during aging, what's causing it? And that's where I also looked at peripheral insulin tolerance to see if that's the issue. That's not the issue. And then just start with the big picture physiological, then to go to the beta cells that could be the issue. So I just went following what I read online through the literature and others have done. Just try to figure out how is the lack of TFF2 affecting the islet function. That was what I was trying to figure out towards the end of my thesis. And towards the end, I with the bulk RNA-seq that we got, we were able to pinpoint it to one factor, but we still didn't fully address that. So that's still in the works for future directions.
Great.
Teresa, do you want to wrap up? Are there any other key points or thoughts you'd like to leave us with?
I guess that was pretty much that we have covered, right? Jose?
Yeah, but what about the future?
Yeah, what do you want to do in the future then? And is
That's
this
the
going to be
question.
Teresa or Jose's future
Jose is
experiment?
gone. Very sad. Yeah, we are actually following up his work right now in the lab. So there are other lab members interested in looking at, for instance, what we discussed that the TFF3 has been the major family members that was compensated. So we now is in the effort of trying to generate the mice that has the TFF3 knockout as well. So we want to ask whether the double knockout of TFF2 and TFF3 in the pancreas will cause an earlier phenotype in terms of the glucose intolerance in mice. Another direction that we wanted to look at is that because we know TFF2 is important to support the beta cell function over the aging. So what about in more drastic metabolic stress conditions, such as feeding the mice with high-fat diet, high sugar diet? Does that accelerate the glucose intolerance phenotype in those knockout mice? So that's currently something that we're doing, we're testing, but we do not have the answer yet. Hopefully there will be something interesting coming up.
And Jose, what are you doing moving forward? What are you working on now?
Yeah, so I mean, TFF2 really dominated my life during grad school, but it really, because it was all I knew from it was from the gastrointestinal field. I was really interested in the small intestine and the cribs, so it kind of allowed me to think about transitioning to the gastrointestinal field for my postdocs. So when I was looking for postdocs, I prioritized looking at labs that do stem cell research and in terms in the context of the gastrointestinal tract. And I found Dr. Omer Yilmaz's lab at MIT in the Koch Institute, and they study colorectal cancer. So now I joined his group, and actually I'm studying the intestinal stem cells and in the context of cancer development. So I'm pretty excited to do that. But I was inspired by TFF2, who introduced me to the intestines. So it was a nice little journey that I went through. But in general, I'm like Teresa knows, I always say this, but the TFF2s are really cool because there's not much we know about them. They're highly stable proteins that can resist very harsh conditions, digestive enzymes, high temperature. It is because of the structure that they have, the triple domain structure that makes them unique proteins, and they're really cool. And they're really regenerative. They can bind to EGFR receptor, CXR4, which are in a lot of tissues throughout the body. So they have tremendous therapeutic potential that other groups from different fields, the skin field, could explore for regeneration purposes. So I'm really excited. I hope this paper and our work can help inspire other people that study TRFL factors and really think about how we can use them for therapeutic purpose. Because there's only a few folks in the intestinal field that are trying to use them, or also for oral injury treatments, but there's not enough out there also that can consider. And we're excited to see what is the receptor that the beta cells express also that respond to TFFs. That's also an answer. So we leave it open to our field to also help us explore and figure it out. I think there's so many unanswered questions that makes this the field really exciting, and also it's now really introducing the idea that there is exocrine communicating to the islets to help regulate them. So we're hoping that this could also inspire other folks to look into other factors that could be involved between both compartments and communicating. So I'm taking a break from the pancreas field, so we'll see if I go back. But for right now, I'm happy to learn about what's going on in intestines. But I always keep in mind the intestines are near the pancreas, so there has to be some crosstalk that I'm always thinking about. And yes, I'm really excited to continue the research that I always like to learn. So I'm just very excited, and I really had a great experience with Dr. Ku's lab. So she really helped me prepare. So thank you, Dr. Ku.
That's awesome. It's great to hear the excitement in your voice
Yeah.
and fun to see your great relationship.
So as we close, Teresa and Jose, we really want to thank you for joining us today, for coming to share your expertise and to discuss your recent work on TFF2 and its role in pancreatic exocrine to endocrine development and function. I encourage our listeners to read the full article. It will be in the September issue of Diabetes.
Thank you for having us.
Yeah, thank you so much.
Next, we're going to discuss one of our feature articles from the September 2024 issue of Diabetes, which is titled The Longitudinal Effect of Diabetes Associated Variation and TCF7L2 on Islet Function in Humans. Today, we are joined by corresponding author Dr. Adrian Vella, who is professor of medicine at the Mayo Clinic in Rochester. Welcome, Adrian, and congratulations on your paper. And thanks so much for being a part of this month's podcast.
Thank you, Darlene. It's a pleasure to talk about it.
Yeah, Adrian, we've known each other for a couple of years, but it's always great to see you, usually by Zoom, unfortunately.
Yes. I have the distinction of being chair of a study section, which only met face-to-face once.
Oh,
So, Adrian, we like to start these conversations with an icebreaker. And before we get started, I thought, maybe you could give us a fun fact about yourself that has nothing to do with science.
Just thinking about it off the top of my head, a few years ago on Delta, because that's where I watch films when I'm flying. That's probably the only time I watch it. There was a documentary, a very good documentary about Luciano Pavarotti. And apparently in the 70s, he cut an advert for MasterCard, I think it was. He basically said, I sing to millions, I'm known around the world. And it cuts to him at home with his daughters and his wife. And he said, but at home, I am nothing. And I thought, I know the feeling. So my two daughters and my wife will be chuckling if they hear this. But anyway.
That works. That works.
all right. Let's talk some science now. So the first thing we like to do is give our authors an opportunity to highlight any other contributing authors or collaborators for this work.
The authors are all listed on the article, and there's the usual list of suspects. So my Italian collaborators at the University of Padova, Claudio Cobelli and Chiara Dallaman. And then there's my long-suffering statistician, Kent Bailey, who's on it. There's my close collaborator, Aoife Egan, and Kalpana Muthusamy, who's new to Mayo, and this was her first project. The first daughter, Maya Zaini, is now doing a family medicine residency. And then there are two summer students, Anisha Ramar, who was here for the summer. This was last summer. And the other person who was on the authors list shares my last name. She's my niece, and she was visiting from the UK for the summer. She's hoping to get into vet school, actually. She should be hearing any day now. And then, of course, I can't forget Marcello Laurenti, who basically came here as a PhD student from Padova and ended up becoming a very close collaborator. So there are a lot of people to recognize, basically, but all of them were essential in their own way to the paper.
Yeah, science is definitely a team sport.
Absolutely.
So why don't you start by giving us an overview of your research program and then kind of how that led to this particular project?
Ultimately, you could say that I'm interested in why people transition from pre-diabetes to diabetes and the factors which drive that. So if you live in Olmsted County, if you're fasting glucose, Olmsted County is the region around Mayo Clinic, okay? If you live here, if your fasting glucose is above 110 milligrams per deciliter, your 10-year risk of diabetes is about 40%, which is high. But you can flip that number on its head, and 60% do not develop diabetes, okay? And so my interest is in trying to understand the factors that drive this resilience, quote unquote. Now, genetics basically came to be used as a probe that I could use to try and test hypotheses as they related to islet function. So in reality, my lab is trying to use sophisticated in vivo techniques, as sophisticated as we can make them, to try and understand why islet function fails in people who develop diabetes and whether there might be early markers of failure. That's ultimately what we tried to do. So initially, when I started, my focus Actually, I started out as being somebody interested in GLP-1 physiology originally. That was my original work as a fellow in Bob Rizza's lab. And then subsequently, I turned to insulin secretion, you could say. And then, because of an experiment that we'll highlight shortly, ultimately, we also went down the glucagon tract. And because things come full circle, there's been increasing realization that the alpha cell makes things other than glucagon. And so now we're back to GLP-1 acting in a paracrine fashion within the islet and perhaps affecting function in a way that might lead to diabetes.
You're tugging at the heartstrings of both of these co-hosts.
Exactly, of course.
So that's a great introduction. Getting to the article, can you maybe introduce for us TCF7L2, right? What is this gene? What does this gene product do? And why is that risk variant important? What does it change in the protein? Some of those questions.
What a mouthful it is, isn't it? TCF7L2, especially when you try to type it, because you're typing in italics, and then you've got to capitalize the L and then the 2. But anyway, I think about this all the time, every time I have to type it. So TCF7L2 is a transcription factor, and it's part of the Wnt signaling pathway. It was actually a relatively obscure thing. The funny thing about it is that it was first described less than a year before it was associated with type 2 diabetes. And at the time that it was described, it was called TCF4. And then, funny enough, within 6 months, so exactly in the middle between that first publication and its association with type 2 diabetes, the whole transcription factor family got reorganized and renamed. And so it became this mouthful, TCF7 like 2, rather than TCF4 as it was originally. And it was described as the transcription factor, which was necessary for proglucagon gene expression in intestinal L cells. So when it was associated with type 2 diabetes in an agnostic fashion, as we'll talk about in just a second, the hypothesis was that the predisposition was GLP-1 mediated, which we subsequently spent a few years showing that it's actually not GLP-1 mediated. But interestingly enough, it took us down the direction we're sort of talking about today. But there you go. So that was the original description. And then, Struve and Grant published the seminal paper, which brought genetics of type 2 diabetes firmly into the modern era, showing that, there's this obscure gene where originally it was a genome, it was a genome scan, which showed this marker that was associated with type 2 diabetes. And then subsequently they did fine mapping and it came down to 2 SNPs. And then a year later, they actually showed that linkage disequilibrium broke down in certain ethnic groups, and the risk SNP was RS7903146. Ask me how I know that off the top of my head. The interesting thing is that the SNP is intronic, and nobody quite knows how it predisposes to diabetes. Okay, it might change. It might introduce splice variants. And the problem is, TCF7 knockouts, for example, have different phenotypes. There's a lot of controversy. And again, it illustrates how, these subtle variants, which have probably very small effects on gene function, you really can't replicate how they affect human physiology by knocking them out in a rodent, because that's a completely different effect. So that's the issue, I think. And in the case of TCF7L2, we're relatively lucky because to date, it remains the common variant with the strongest effect size on diabetes. So it is within the realm of the possible that you can do human studies, certainly within the life of an RO1, which is, of course, another significant factor, okay? So it's, the odds ratio of one allele is 1.4, something like that, okay? Nowadays, the 200th SNP associated with type 2 diabetes has an odds ratio of 1.00 something. I exaggerate slightly, but essentially you're not really going to reliably detect an effect with 200 people with the diabetes allele and 200 without.
Right.
In those sorts of SNPs. Whereas TCF7, we're lucky because it's been fine mapped, and we know that if that SNP RS7903146 isn't the etiologic variant, it's indistinguishable from the etiologic variant by linkage disequilibrium. And the second part is that the effect size is decent enough that you have a chance of getting it, of finding something with a reasonable effect size.
So tell us about the methods for the current paper. Where are you finding all of your subjects, and how did you study them to get the result?
Yeah, so that's actually a great question. In the beginning, when I started out, I naively thought that we'd basically put up a sign saying volunteers wanted, we'd get people in, genotype them, and then choose people. By design, we've always gone for the people who are homozygous for the disease-associated allele and those who are homozygous for the disease-protective allele, because like that, you're maximizing phenotype attributable to the genetics. The problem is that in the North American population, the prevalence of people who are homozygous for the diabetes-associated allele is somewhere between 10 and 15%. In Olmsted County, for some reason, at least in the early days of our recruitment, we'd screen 100 people to find 5 who were homozygous for TCF7L2. So, and then I was lucky because we had a biobank that had been established in the mid-2000s. And the idea of that biobank was really to provide a pool of controls for genetic association studies for oncology type associations. It was never really designed to recontact these people and have them come back to participate in studies. But thankfully, with a little bit of persuasion, we actually were allowed to genotype these people and then match CCs and TTs based on their weight and their age and their sex, and then ask for permission to contact them. And actually, the biobank would contact these people on our behalf and say, is it okay if they contact you? We were lucky because we were able to recruit reasonable amounts of people in a reasonably short period of time that way. And that method has helped us since, we have established cohorts, the people who we've contacted before, and there are still people who we've never contacted in that biobank that we could contact if we wanted to.
Nice.
So let's move on to the results. Can you tell us a little bit about what you found regarding TCF7L2 and its regulation of glucagon dynamics, and then those associations with the onset of diabetes?
So if I can, I'll give you a little bit of historical background. The first cycle of my studies in TCF7L2 were actually designed to try and determine whether it affected response to GLP-1, whether secretion of GLP-1 or response to infused GLP-1. And those were negative. But as part of a study assessing response to infused GLP-1, the people who were homozygous for the T allele actually failed to suppress glucagon in response to a hyperglycemic clamp, okay? That was an observation that we made. We stated it. It was a relatively small number of subjects, and it was unexpected. We didn't make much of it, and we ignored it. Subsequently, we set out to do this experiment where one of the things that worried me at the time was that when we use the minimal model as a measure of beta cell function, we relate the insulin secretion to insulin action. It does have a hyperbolic relationship. My concern was that this hyperbolic relationship might differ within populations. Certainly, we know that the relationship in people with type 2 diabetes is very different to that in people who have normal glucose tolerance. So we knew that. And I thought, the TCF7 gene is a perfect probe to try that. In my naivete, I thought we were going to describe two hyperbolic relationships. One would be CC and the other one would be TT, C and T being the alleles respectively. So we designed this experiment where we would study people in sort of the basal state. And the second experiment would be we'd make them acutely insulin resistant with raising free fatty acids using heparin and intralipid. And, the hypothesis crashed and burned. The hyperbolic relationship did not differ at all between the two groups in response to that. But clearly in the TT group, glucagon suppression was very, very abnormal. And the funny thing was that all of these people had undergone a 2-hour OGTT. So we actually went back and did glucagon in the OGTTs, and sure enough, they did the same thing. So we had, 3 study conditions where the people who were homozygous for the diabetes-associated allele actually had an abnormality of glucagon suppression in response to oral glucose. So that was really when we thought, well, it seems that TCF7L2 seems to act through the alpha cell as much as it acts through the beta cell. We replicated everybody else's finding that it does affect insulin secretion, but this was novel. Now, the interesting thing was, at the time, this was a cross-sectional study, right? So you're showing that these people have impaired beta cell function and impaired alpha cell function. The question is, which comes first? Or which worsens more than the other one? Is it an innate defect, which is just, expressed over time as people become overweight, et cetera, et cetera? So that was the genesis for the study that we're discussing today. It was really a simple study. We do an oral challenge one day and then wait 3 years. Again, that's the life of an RO1 that you could reasonably expect, right?
Yeah.
And, fingers crossed, we can see some effect of some sort. Of course, in addition to, delays in recruitment, et cetera, there was a small infectious disease thing that happened in 2020, which kind of might have had some effect on the ease with which we could conduct the study, but we got it home, and it seems to suggest that at least the early change is alpha cell dysfunction.
Very cool.
Let's talk about, it seemed in your discussion you'd mentioned this previous data and your current data, and there's kind of this mismatch, right, between what you would predict to happen to glucose under these changing hormonal conditions. And so can you tell our listeners a little bit more about what you think's going on there?
Absolutely. That's a very good point. And it's a point which actually isn't surprising. And I'll tell you why. When I started out in Bob's lab in 1998, I was a first-year endocrine fellow, and they were just putting the finishing touches on a paper which got published subsequently. The first author was my colleague, Pankaj Shah. And they showed that glucagon abnormality, a failure to suppress in response to a meal, is only significant in terms of affecting endogenous glucose production and hyperglycemia. Really significant when beta cell function is basically impaired. And to a certain extent, that remains correct. The only problem with that experiment was that it was conducted in relatively young, I think they were in their late 20s on average, and BMI was in the 22 to 24 range, something like that, at least median, okay? So the message was the alpha cell isn't really important if the beta cell, to glucose tolerance, if the beta cell is intact. And so the thought that the alpha cell could be an early mover in the pathogenesis of prediabetes was sort of, in my mind, that wasn't really an issue.
Yeah.
Right? However, if you've got a gene which to date remains the strongest association with type 2 diabetes, where at least half of its effect is mediated via alpha cell dysfunction, you of course have to question that premise, right? Because a gene association which affects the alpha cell implies that probably there's an early change there that is relevant to the pathogenesis of diabetes. That's one important point. The other important point is when we replicated Pankaj's experiment in an older cohort that was overweight, then the effects are far more marked on glucose tolerance when you impair the alpha cell. Actually, we didn't really impair the alpha cell. What we actually did was we replicated conditions where the alpha cell isn't suppressing, okay? So obviously we exaggerated the effect of what we normally see. But at the end of the day, what it comes down to is, in my mind, okay, is that these people still have relatively intact insulin secretion. Moreover, and more importantly, they're generally insulin sensitive. Most of them had normal fasting glucose, and the vast majority were normal glucose tolerant, okay? So having a glucagon concentration which is abnormal in those circumstances, to me, is more, it's a marker that the islet is misfiring, so to speak.
Yeah.
People who know me well know that I start making car analogies very, very quickly, okay? And the analogy I'm going to make here is, it's like your brakes are binding on slightly. And perhaps because the engine is having to work a little bit extra to overcome that, you're perhaps predisposing for failure. Now, that's assuming that our long-standing, generally agreed upon hypothesis that overwork of the beta cell is necessary for failure down the line holds true, which isn't actually a given either. But that's the analogy that I try to think of when I'm asked a question exactly like you asked me.
Can I ask a off-script follow-up
Yeah.
to that? I'm curious. So if you knew you had a patient that had the risk allele and they were still pre-diabetic, would you treat that pre-diabetes differently?
That's a great question, Darlene. No, I wouldn't actually, okay? Because on an individual level, knowing what your TCF7L2 status is has such a small effect on your overall risk of diabetes or the pathogenesis of diabetes that it doesn't really help you individually. As a group, yes. And if we knew for sure that there were ways of changing alpha cell dysfunction, that would be a very important experiment, a very important hypothesis to see if we could affect outcomes and what numbers we would need to affect those outcomes. But I think here we highlight the beauty of genetic association studies, but at the same time, their limitations. So it's illuminated a whole pathway for us, but it doesn't really have applicability to you or me, whether we know what our TCF7L2 is or not, status is, genotype, that should be.
Adrian, that was a great discussion on your paper, and we want to thank you a lot. As we close, are there any key points or thoughts that you'd like to leave us with, or maybe give us a hint as to where you're going with it?
I will certainly give you more than a hint as to where we're going and what my thoughts are. And this was actually, so we've at ADA just a couple of months ago, we had a poster where we actually reanalyzed the data in terms of, I don't care what the genotype is, I just want to see how alpha cell function at baseline might have predicted the progression. And sure enough, it does, okay? It does predict progression. Of course, this is a bit of, the codependent variables because most of the people who had abnormal alpha cell function had the TCF7 genotype. But somebody, a good friend of ours at the poster, pointed out that that group had a higher peak post-challenge glucose at baseline. The people who progressed, the progressors, had a higher peak glucose, despite having normal beta cell function, as measured by the minimal model. And I've actually been on a 5-year attempt to try and develop an alpha cell model, an alpha cell module of the minimal model over and above the beta cell. It's proven to be a lot more difficult than I imagined. But even in this cohort, you see, glucagon concentrations per se don't really differ between the two groups. but they're occurring at different glucose levels. So we've had a good way of relating the glucagon secretion at any given moment with the glucose, either at that moment or depending on what the lag phase is, which we don't quite understand as yet, the glucose 10 minutes before or 15 minutes before, where it was necessary to signal to the alpha cell to suppress. So we're actively working on these things. We've been working on these things, but it's easier said than done.
Yeah.
Right.
Very cool. Adrian, we want to thank you for joining us today and sharing your expertise in discussing your recent work on TCF7L2 and its role in glucagon dynamics. I encourage the listeners to read the full article in the September issue of Diabetes.
Thank you all.
Welcome to another edition of Sweet Talk. I'm Kevin Williams, and today we welcome Dr. Matt Weber from the University of Notre Dame. Dr. Weber was awarded the American Diabetes Association Pathway to Stop Diabetes Accelerator Award in 2019. His research combines material science and engineering to develop smart materials that can sense blood glucose levels. These materials are designed to release insulin when glucose levels are high and glucagon when glucose levels are low. Dr. Weber contributed an article to the July edition of Diabetes that highlights this work and the impact the Pathway Award has had on his research. We are excited to have him with us today to talk more about this exciting topic. Welcome, Dr. Weber, and thank you for joining us.
Yeah, thanks, Kevin. Happy to be here and happy to have an opportunity to discuss our research.
Great. Let's get started with an icebreaker question. If you had not pursued a career in science, what profession do you think you might have been drawn to instead?
Yeah, I always and still am somewhat drawn to the idea of really working in a translational capacity and maybe like a startup company or something like that to bring technologies to people. We do aspects of that in academic research. I think what I would have liked about that is the kind of constant need to kind of be hungry and continue to strive to even survive in some cases in those types of environments. I realize I
Absolutely.
say that from one of the most comfortable positions in the world, that of a tenured professor at a financially stable private university. But I do think it would be kind of a fun challenge to kind of be in that really kind of fast-paced, kind of cutthroat environment of a biotech startup scene, as a scientist or as a leader in some capacity there. And so probably if I hadn't done this, I would have done something like that. So still within science, but probably kind of a different manifestation.
Perfect. And I gave a brief overview of your work in the introduction, but maybe you can tell us a little bit more in detail about your research and how you got involved in researching diabetes.
I don't have a personal connection really to type 1 diabetes, thankfully. I'm not diabetic. My children are not diabetic. I don't really have any type 1 diabetics in my family. And so that's one of the questions I often get is, why did I get involved in this line of research? And it's actually kind of simple in that, I wanted to go to work in the labs of Bob Langer and Dan Anderson at MIT for my postdoctoral fellowship training. They had received funding at that time from the Helmsley Charitable Trust to do work in glucose responsive insulin delivery. And that was the opportunity that presented itself, was to come in and help lead some of that work in their lab and develop new technologies for glucose responsive insulin delivery and in treating type 1 diabetes. And so that was really it was just a really interesting and challenging problem that allowed me to use my skills in synthesizing new materials and doing molecular engineering of small molecules and other sorts of systems in order to contribute new technologies to solving what really is still unaddressed, which is, how do you make diabetes therapies more responsive to the exact time and level needed, right? And so can they sense blood glucose and in so doing tune their potency or tune their availability in a way that could more accurately and precisely control blood glucose the way a healthy, normally functioning pancreas would. And so this was really it was just an opportunity to go work for people that I wanted to train with and work on a really, really challenging problem that I think really continues to challenge us to this day.
Yeah, it's great. You're working with these polymers that, when packaged with insulin or glucagon, can then release these hormones in a glucose responsive manner. I'm curious about, how do you find these materials that you're using for this purpose? Do you repurpose materials that are used for other applications? Are you synthesizing entirely new ones? What's the strategy for the development of these polymers?
Yeah, almost all of our systems are brand new synthetic approaches to this strategy. We're not really repurposing already approved or already used biomedical materials in any meaningful way. We're synthesizing new things, and so it's often very trial and error. It's things that we draw up on a whiteboard or on a sheet of paper that we think could work, and then we figure out ways to make something that can do what we want it to do. And so this begins really with a lot of chemistry, a lot of material synthesis and characterization. And then even in my own lab, we do, everything from the synthesis all the way through in vivo testing in mice in our lab. We don't have really any collaborators in any of these spaces. We've just kind of developed the ability to synthesize, screen, test, and evaluate different technologies across the full spectrum of at least research development or academic research development. I think for some of these technologies, we've been partnered with people to do large animal studies, like in pigs. We don't have that expertise. We do need experts that can handle and monitor and manage diabetic swine. But up until that point, we really do all of the idea generation, synthesis, development, and validation in our lab.
That's really great. I'm curious too, the focus appears to be on glucose sensing. I'm curious, is there a broader focus for the applications at play here for the polymers? Do you see this research as potentially applicable to other diseases that might have specific biomarkers? Or have you considered using this technology in other disease areas?
I think at my core as a scientist, what I really like to do is make dynamic and stimuli responsive materials, just generally speaking. Glucose responsiveness and dynamic materials is one really useful application of the technology, but I think there are others. And so we've thought about making dynamic materials that could be, injectable depots for all kinds of different drugs. We've thought about these in conjunction with delivering cells or with delivering protein therapeutics for treating different diseases. We've thought about these now as making new matrices that could more accurately recreate or capture the cancer niche within, what cells experience in terms of a dynamic matrix environment in cancer and these sorts of things. And so certainly we're broadly interested in a lot of stuff, but it really at its heart comes down to a fundamental interest in making dynamic and stimuli responsive materials at a molecular scale.
Yeah, it's really fascinating. With this technology, has any of it made it past preclinical stages into clinical trials, maybe into the clinic? What stage of development do you see us at in terms of seeing these materials being used in patients?
Our own work has not advanced beyond, large animal studies in a couple of examples, and a lot of it still is in small animals, mice and rats. And so we're still in that process for a lot of these technologies. I think the runway to get something into even clinical trials is quite long and very capital intensive. And so we want to make sure anything we move forward with is really well engineered and well vetted and ready for translation. And so nothing we've done has so far advanced to the stage of testing in humans, but we're really close on a couple of technologies. Some technologies that that we'd invented and had been licensed by a startup company are kind of knocking on the door of at least early stage evaluation in humans within the next year or so. And then we're working now on some other technologies in a little bit earlier stages, but starting a couple of companies and, raising capital to try to push a couple more technologies in insulin and then in some therapeutic peptide work that we've been doing to the next phase, which would hopefully lead to testing and evaluation of efficacy in human.
This is really a fascinating topic. I'm curious, what excites you the most? What new directions do you see diabetes research heading towards?
I'm really sensitive to the need for better insulins. I think that especially in type 1 diabetes, if you had insulins that could provide a better profile of activity, you could mitigate a lot of the challenges in both the acute and chronic blood glucose control that are faced by diabetics. And so we think about insulin that could be very long acting, but then also could respond to, meal times or elevations in blood glucose with enhanced potency. But then in so doing, you also need to think about engineering these same insulins to have the shutoff mechanism to prevent the really scary acute stuff, which is the hypoglycemia, right? And so I think that that's a really challenging problem to try to engineer something that can really function with both an acceleration and a shutoff in function corresponding to real-time changes in blood glucose level. And it's really unlike treating any other disease there is. If I want to design something to treat cancer, I don't need that kind of precision in terms of an on and an off switch for activity, right? The kinetics of the disease are so specific to associated with meal timing or, hypoglycemic episodes that may arise when somebody's sleeping or, other sorts of things that diabetics know all too well. And you have to have a therapy that can meet those demands. And I think that that's really unlike treating any other disease. I do think that increasingly what we're seeing with like GLP-1 receptor agonists and related peptide therapeutics for type 2 diabetes and obesity and these sorts of things are going to be paradigm shifting in terms of our ability to treat those diseases. And so whether insulin becomes as prevalent of a component of treatment in type 2 diabetes, I think it looks likely that it is going to be less commonly used moving forward. I think that with some of the remarkable advances just in recent years with drugs like Ozempic that probably have even household recognition now and around a lot of most circles. I think that that's going to really change the course of treating type 2 diabetes. But type 1 diabetics are always going to depend on insulin until such a point when there is maybe a cell-based cure, which has been on the horizon for a while. They're always going to depend on insulin. And so we need to make better insulin for these people.
Yeah, the precisionness of the dynamic range that you're talking about is really important as a criteria in the treatment of diabetes. And the research you're working with does appear to be on that transformative path. So it is really exciting to talk about this topic. Thank you, Dr. Weber, for joining us today. And thanks to our listeners for tuning in to this episode of Sweet Talk. Be sure to read Dr. Weber's article titled Engineering a Pathway to Glucose Responsive Therapeutics, available in the July 2024 edition of Diabetes. And as always, please like, follow, and subscribe for more Diabetes Bio content.
Thanks to all of you for joining us on the Diabetes Bio podcast. We hope that you're enjoying our discussion of the work that the Diabetes Journal is publishing each month. The feature articles we discuss in each episode are freely accessible at diabetesjournal.org/diabetes and are also listed in the episode notes for your reference. As with any podcast, it really helps us if you like, rate, follow, and subscribe to the podcast. And if you have any feedback for us, we'd love to hear that as well. We look forward to presenting to you each month.